So in today's class we are going to study TS of Young Anther. This diagram what we are going to study now with the structure is very important for board examination, where it will be asked for 5 marks. In today's class we are going to study how to draw the diagram and what are the parts of the TS of Young Anther and how it is producing the pollen grains. So now let us draw the diagram first. Along with the diagram we will learn. So the very firstly we can draw similar to a butterfly structure, Okay, fine. Next, which is the next layer? Next layer is endothecium. So in anther there are 4 walls. So we wrote 2 walls now. Another 1 wall I am drawing now. So 1 more wall. Okay. So then comes the sporogenous tissue. So the outer wall is epidermis. This epidermis is single layer outermost single layer and protective in function. Then comes the second layer, endothecium. The first outermost layer is epidermis. Then the next layer is endothecium. This is thick, made of cell wall with pectin and cellulose, protective as well as helps in dehiscence. So when the pollen grains are produced, that pollen grain should come out of the anther. So this endothecium helps in breaking of that wall. Then the third layer is middle layer. 3 to 4 layers. Okay, 3 to 4 layers. Their thickness, leaving and also helps in dehiscence. Okay. So surrounding this is the sporogenous tissue, where the sporogenous tissue is present, and surrounding this sporogenous tissue, where the sporogenous tissue is present, that sac is called microsporangium. So in our NCERT they are telling that in TS of Young Anther, microsporangium is almost near to circular in outline. This cells, the sporogenous tissue present in the microsporangium is covered by a nutritive cells called tapetal cells. tapetum or tapetal cells. The function of tapetum is nutritive, okay, bi or multinucleate, since they are always producing nutrients for the developing spores, right? Then dense cytoplasm, okay. So that is with the tapetum. So next we will go with this enlarged part in the next page. And one more, as we studied in the previous sessions, here there will be one connective tissue here. Okay, what is this one? This is a connective tissue which holds everything in place. So in the next page we are going to study the enlarged portion, just how to draw the diagram. Write a inverted C, then our sporogenous tissue present, that is in microsporangium. So this is the outer epidermis, one cell thickness. Okay, then comes our endothelial layer. Okay, then comes the middle layer. The function of middle layer is also dehiscence. Okay. Then the next is tapetal cells. With dense cytoplasm, multinucleus. Okay, and inside the microsporangium, what is present? We have a sporogenous tissue. So this is sporogenous tissue. So labeling once again we can do here. Okay, so how we will draw? The outermost is epidermis, followed by an endothelium, then comes middle layer, and tapetum. So tapetum is surrounding the sporogenous tissue. So now this sporogenous tissue contains a specialized cells called archisporial cells. This archisporial cells develops and differentiates into pollen mother cells. Okay. This pollen mother cells undergo meiotic division to form pollen grains. What are pollen grains? They are the male gametophyte. The process of formation of male gametophyte from the pollen mother cells involving meiotic division is called microsporogenesis. The process is called microsporogenesis. So in the next slide we will be studying the microsporogenesis. Now microsporogenesis, the name itself is indicating us that the formation of micro spores. How? Using pollen mother cells. These pollen mother cells are diploid. So they are still diploid. So undergo meiosis 1 to produce two haploid cells. Okay, two daughter cells, and both of them are n in number, haploid in chromosomal number. So this compulsorily undergo, both the daughter cells compulsory undergo meiosis 2 to produce an microspore tetrad, which are all present intact in a tissue called callus. So this is the microspore tetrad. The tapetal cells produce an enzyme called callase. Which cells the same tapetum, okay, which are also nutritive in nature, producing callase enzyme, which helps in removal of digestion of this callose, releasing the pollen grains freely. These are microspores, also called pollen grains. Each pollen grain is now nourished and secreted with sporopolanin and other important chemicals, organic materials, and this is called pollen kit. Pollen kit helps the pollen grains to protect themselves. Okay, so that is with the microspore or pollen grain. The entire process is called microsporogenesis. Fine. So ಇಲ್ಲಿ stop ಮಾಡಿ ಒಂದು ನಿಮಿಷ ಏನಾದರೂ ಪಾಯಿಂಟ್ ಮಿಸ್ ಆಗಿದ್ರೆ ಹಾಕ್ಬಿಡು. Okay. So now the important function of tapetum, as we studied in the previous slide, is nutrition. So they are providing the nutrients for the developing pollen grains. So microspore mother cells are there, which divide meiotically to produce two daughter cells which are haploid, and both of the daughter cells compulsorily undergo meiosis 2 division to form the microspore tetrad. And this microspore tetrad is surrounded by the callose. Callose is the coat that is produced by the tapetal cells. And the same tapetal cells also producing callase enzyme, which dissolves or digests the callose, releasing the pollen grains freely. And each pollen grain is now surrounded by a pollen kit, and this pollen kit is again produced by the same tapetal cells. So this pollen kit is containing proteins, accessory coat, also lipids, which overall protect the pollen grains, and pollen kit is sticky. Pollen coat is sticky, sometimes dry, as we see in wind pollinated flowers. So fine, that is all with the microsporogenesis.